start.
Ya kunde tusha dhavala ya shwet padmasana ya brahmachyut shankara prabhuti देवै सदा वन्दिता देवै सदा वन्दिता. Good afternoon, everybody. I welcome all of you, myself, Dr. Chetna Rajaguru, from MVM Science and Home Science College, Microbiology Department. I heartily welcome all of you on the platform of this 6 days lecture series. Here we are going to start national level lecture series on inspiring aspects of microbiology and biotechnology for 6 successive days from 2nd November to 7th November 2020. We are very happy to share with you that an overwhelming response we obtained from all over the country. We found 585 participants already registered on the last date of October month. Now, I would request our young and energetic principal, Dr. K.J. Ganatra sir, to say a few words to inspire us. Dr. Ganatra.
Thank you, Chetnavan. Respected Dr. Kale sir, the trustees of our college, Dr. K.K. Kakkar, Meenaben Pandi, Madhuben. Though Meenaben has joined us, the Kakkar Sahib and Madhuben probably have not yet joined. But anyway, the faculties of the microbiology department of my college, Dr. Umrania, Dr. Chetnaben Rajguru, Juhi, my other colleagues in the college, especially I would like to mention Pankaj Chovatia, who has made these online sessions possible, Dhatri Ben, and finally, the participants who have joined or will be joining from across the nation. I welcome you all. It's my pleasure to welcome you all at this lecture series, inspiring aspects of microbiology and biotechnology to be connected, to be addressed by the eminent scientist, Dr. Sharad Kale. The participants or the students are familiar with this subject. They may be pursuing their studies or courses at the UG level or PG level, or some of them may be research scholars. So primarily, you may be familiar with this subject being a student. But the object of this lecture series is intended to create a better insight into the subject, and which will perhaps make you more thought-provoking about the aspects of microbiology and biotechnology. So the main objective of this whole lecture series that is going to be organized is to inspire you with the better thoughts pertaining to the subject. So on behalf of Sri Jalaram Utkarmani Mandal, Matu Shivirbhama, Mela Science and Home Science College, I take this opportunity to welcome you all, and thank you, Dr. Kale sir, for having accepted our invitation to conduct this series. I would like to urge all the students to utilize this opportunity for a better understanding of this subject. So thank you very much, and wishing you all the students and participants for a positive outcome at the end of the series. Thank you, sir. Thank you all.
Thank you very much, sir, for your inspiring words. Now I will request our head and professor, Dr. Valentina Umbrania, to introduce the speaker to all of us.
Hello everybody. Namaste, Jai Jalaram. Good afternoon and heartily welcoming all of you. Today, the theme of our seminar, as we know, that we want to have inspiring aspects of microbiology and biotechnology. We have to think from the out of the box and to start thinking about the innovative ideas, about the experiments, and about the knowledge in the field of micro and biotechnology. Dr. Sharad Kare sir is from a small town, Nagar, near Pune, and he did his MSc from Pune University and joined BRC in 1977 as a scientific officer in biology division. Later on, he served as a head technology transfer division BRC and then he retired. He has several national and international publications and has visited laboratories all over the globe. He has written several popular science articles and has held awareness programs for students, farmers, and even housewives. He is the best known person for the development and promotion of Nisargarund, that is the biology of the production of biogas, which has been licensed more than 100 licensee and several plants are functional in the country, including one at the Delhi Secretariat. He has been awarded Civilian Award Padma Shri for the contribution to the society. He is a part of several committees which contribute to policy evolution in the field. He is a teacher at heart and an inspiration to all of us. He has great love for Mother Earth and he works constantly for the care of our environment. Nisargarund, Nisargadut, and Nisargalakshmi are the concepts he has introduced, which creates wealth from the waste. Let us now listen Dr. Kare sir and enlighten our pathway in the field of biological sciences. Thank you so much, sir, for being with us and sparing time to all of us for this wonderful occasion. Thank you, sir. I welcome all of you here for this 6 days webinar series. Thank you.
Thank you very much, madam, for your very interesting introduction of Kale sir. Now I request Dr. Kale sir to begin his valuable talk and enlighten us on the knowledge. Please, Dr. Kale sir. We are heartily
I think sir is
welcome.
not online.
We heartily welcome all of our dignitaries, faculties, and researchers on this platform. We are very happy to see all the students and faculties together on this occasion. This is the first time where we are meeting online for discussing inspiring aspects of microbiology and biotechnology. Kale sir is a very experienced scientist. He can explain all about degradation and utilization of garbage and waste material into very valuable things. which can be utilized for purpose of agriculture, and also it can be used for some other purposes by using scientific technology. So we are soon going to see him, and he is going to enlighten us.
Sir is present now.
Welcome, sir.
Thank you. Shall I start?
Yes, sir.
Okay. Thank you very much for giving this platform. I am Sharad Kale. I will be speaking to you for next 6 days. I am sure all of you will be wondering that without giving any specific topic, without talking, without telling that 6 days what I am going to speak, and what are you going to listen? That also you might be in a rather dilemma कि ये 6 दिन हम क्या सुनने वाले हैं, except the topic is given, which is rather a very vast and general topic, that is inspiring aspects of microbiology and biotechnology. Well, let me begin with a quote from Sir Isaac Newton. Sir Isaac Newton has said a very famous sentence. I am sure some of you might have already read about it, that I do not know what I may appear to the world. Newton says, I do not know what I may appear to the world, but to myself I seem to have been only like a boy playing on the seashore and diverting myself now and then in finding a smoother pebble or prettier shell than ordinary, while the great ocean of truth lay all undiscovered before me. It's a beautiful sentence that Newton is considering himself as a boy. He is standing on a seashore. The vast sea of undiscovered truth is there. And he is holding a kind of, his attention gets occasionally attracted to a small pebble or to a kind of very different type of pretty shell. So he keeps on observing it. And isn't it a beautiful way of living your life? That you keep on discovering, you keep on looking for some new things. See, when life becomes routine, yeah, you must have experienced in this corona pandemic, especially during the lockdown. जब लॉकडाउन चल रहा था, तब आप लोगों ने ये महसूस किया होगा कि कितना बोरिंग होता है लाइफ, कि आपको कहीं बाहर नहीं जा सकते, किसी मित्र दोस्तों से नहीं मिल सकते, पार्टी को शाम को इकट्ठा नहीं आ सकते, बर्थडे नहीं मना सकते, सारी चीजें जो हम आदी हो गए हैं, ये सारी अगर चीजें नहीं हो तो लाइफ बहुत बोरिंग लगता है. और साइकोलॉजिकल प्रॉब्लम्स जैसे मानसशास्त्रीय समस्याएं ये लॉकडाउन में बढ़ती भी है. आपने भी देखा होगा, घर में अगर इंसान कंटिन्यूअसली 24 घंटा रहे तो झगड़े भी होते हैं. तो ये सारी समस्याएं क्यों पैदा होती है? क्योंकि हम हमेशा कुछ नया चाहिए हमें. जब हम उठते हैं सुबह तो कुछ नया विचार लेके उठते हैं. We have to get up with a new thought. अरे today I will be doing this, today I will be doing that, today I have to really learn something new. Today this lecture is there, tomorrow that experiment is there. So आप सब students हैं, so you should be looking forward in your life. And precisely, I like to say I don't have a magic जैसे जादू की छड़ी नहीं है मेरे पास कि मैं आपको ये पूरे 1.5 घंटे में और 6 गुना 1.5 9 घंटे में कुछ मैं जरूर आपको नई कल्पनाएं दूंगा, नए विचार देने की कोशिश करूंगा, जिसमें से काफी सारी विचार आप आपके जीवन में ला सकते हो. और ऐसे कई आयाम है इस दुनिया में, इस जीवन के, जिसे where you have to keep on looking for the new things and don't expect that every day new thing will come, but every day your mind should be enthusiastic. And that is what probably Newton means, that his mind is very enthusiastic. When he is looking at things in the world, he's always, I remember one of our Marathi authors, his name is Pula Deshpande. He was talking about Rabindranath Tagore. And he says Rabindranath Tagore at the age of 80, he has that in his eyes, these which are very old eyes. But though in those eyes, I can see when he met Ravindranath at that age, Pula Deshpande said, I could see that brightness in his eyes, which I find normally in the eyes of the children. And that is what I expect out of this lecture series, that when this lecture series is going on and when it ends, there has to be some kind of brightness in all of your eyes. It's not that I'm going to teach you, because I know that your classroom teachers or the college teachers are much better teachers than me. So I'm not going to actually explain to you. Maybe I will explain few things. I will explain few principles. I will explain few instrumentations and the good laboratory practices. I will probably introduce you to some good aspects of microbiology, like fermentation, the proper degradation, the environmental cycles. So I'll be discussing a lot about biotechnology and microbiology, but my intention is not to teach you the classical or the classroom subjects which already are being taught to you. Generally, what happens when you are trying to study in your life, the question is what and how to learn. You and what are the normal things which are what is the normal course of learning? There is a fixed syllabus, there are textbooks, and there is experimentation. You call it practicals. I don't like the word practicals really, but experimentation is a better word. The research papers and science communications, you have lecturers, you have field visits, you have seminars, you have tutorials, and you have self-study. You do the notes after understanding the topic. Well, this is, I want to go beyond that. I don't want you to, I mean, I don't want to restrict myself to these syllabus, because I know that the audience which I am having is a rather vast audience. It is from BSc, MSc, even some housewives have joined. So I would like to have a kind of a generalized syllabus, but basically what a student should know, or what student wants, is he wants three things. He wants inspiration, he wants motivation, and he wants an aim to the life. The inspiration generally comes from famous scientists or the books. You know, Dr. Bhabha, he had a beautiful library of his own. And this in the library, there were so many books. Of course, Baba was born with a, I won't say even silver spoon, he was born with a gold spoon in his mouth. So he had plenty of resources. But with those resources, with that kind of library, you know, the way Dr. Baba developed himself, in any rich family, you generally don't find a student or a son in that family to be a good student, because with so much of money backing, they don't really care to study. Dr. Baba was an exception. He, with such a knowledge, which is such a wealth behind him of both the book wealth as well as the money wealth, he decided to be an physicist. And his father was insisting that he should become an engineer. So he made a deal with his father. He said, "Okay, Daddy, if you want me to become a father, I mean, if you want to become a engineer, I will certainly become engineer, and that too. But after engineering, I will like to study physics." So his father was taken aback. He said, "After engineering, I want you to join the Tata Group, so that the Tata Group is looking for some good engineers, and I want you to be the engineer there." He said, "No, doctor, no, father, I will like to become a physicist." So his father then, after all, he was Bhaba's father. He said, "Okay, let me get this thing clear. If you pass your engineering with first class first, that is distinction, and first gold medal, we could get a visionary at the helm of affairs of Department of Atomic Energy. We could get a visionary at the helm of affairs of Department of Atomic Energy, and that's why India has progressed so well in the field of in the field of atomic energy. In fact, Dr. Baba's papers, if you see, the way he has read Einstein at the very early age. Einstein is very difficult to understand, let me tell you that. Quantum physics is difficult to understand, but Dr. Bhabha, being at very brilliant, and because resources were available to him, so that was the inspiration. See, when Dr. Abdul Kalam talks to children, I mean, now he is no more there, but I will always like to bow in front of the photograph of Dr. Abdul Kalam. I consider him as a god. That when he used to go and mix with students and talk to them, it was so inspiring that such a scientist of such a caliber is coming down. When he went to the president house after being elected as president of India, he went to president's house with only 2 bags of books. And when he came out of that president's house after his term was over, he came out with 2 bags of books. So that's a dedication, my dear friends and my dear students, that what kind of personality was Dr. Abdul Kalam? And still at the age of 83, till his last breath, he was talking to the students. The job which was been entrusted to him by the Almighty, you can call him God, you can call him Allah, you can call him Christ, you can call him supernatural power, whatever you want to call. Whatever that job had been assigned to Dr. Abdul Kalam, he made sure that he was doing that job till the last breath. And that is what is called life. That is what is called inspiration. What we have to learn from Dr. Kalam, what we have to learn from Dr. Bhabha is all these aspects of life, and then you will be motivated. The books is another beautiful things which really inspire you. I wonder how many of the children, students who are listening to me have read one microbiology book from page number 1 to page number last. And that is what sometimes really bothers me, that people are losing faith, students are losing faith in their textbook. They are more dependent upon marks. All of us are marks for the education. We don't want marks for the education. We want gyan for the education. We must gain knowledge out of it. It's not for marks. So some people keep it for something for option. My dear friends, there is no option in life. If the doctor says that I had kept heart in option and now you have heart attack, so you look for some other doctor who had taken heart as his subject or his liking, it will be a very difficult situation. In nature, there is no option. In nature, there is no going back. It is always going forward. You can take a step back in your computer chess game, but not in real chess game of your life. You cannot take anything back. And that is what is taught to you by books. I remember one beautiful book. I don't know how many of you have read that. That book is on a medical fraternity. It is written by A.J. Cronin, and the book's name is Citadel. It's such a beautiful book. In that, there is a passionate doctor who has come from poverty and who has come up to the level of MD, and he appears for his MD Viva. And in that MD Viva, he says that there are 2 examiners who are very, you know how Viva examiners are there. I don't have to tell the young MSc and BSc students that how Viva can be very trembling experience or very traumatic experience for all of you. So similarly, a traumatic experience was in waiting for this young doctor, because those 2 doctors which were going to take his Viva were known for their meanness and their very difficult questions which they used to ask to the students. So the first examiner gives him 16 slides, histopathological slides to explain. This candidate tries his best, and 1.5 hours he explained those 16 slides, but there is no change on the face of the examiner. So the candidate is perspiring. He doesn't know whether the examiner liked it or not. And in that confused spirit only, he comes to the second examiner. And the second examiner sees his dress, because he is coming from a very poor family, which is obvious from his dress, and as well as trained face. And then he knows that this candidate is really a very passionate candidate who has studied well. So he tries to make him at ease. And he asks some few simple questions, then very difficult questions, goes on probing. And he knows that the depth of the knowledge of this candidate is very good. So he asks him, he says that I will ask you 2 more questions. He asks that who discovered anaerobiasis. So this boy tells him XYZ is supposed to have discovered anaerobiasis. Then the examiner is surprised. He says, "Why, doctor? Why you say that he has discovered anaerobiasis? He has discovered anaerobiasis. He is not supposed to have discovered." So the candidate says, "Yes, sir. In 6 textbooks, I confirmed that he has discovered anaerobiasis. But still, for this exam, I have to pass my Latin, and because I want my Latin was to be refreshed, I was going through Latin literature, and in that Latin literature, I came across a word, anaerobiasis, which was 1300 years before it was supposed to have discovered by this XYZ." So the examiner is impressed. He says that in this hall, this is for the first time somebody has told me that something which I did not know. And he says, okay, last question. And that last question, my dear students, it's an inspiration to me throughout my life. That last question, he tells him, is that, doctor, what will be your approach towards medicine? You are going to get full marks. Looks like you are getting good marks. You are going into the society as a doctor who is going to take care of patients in the society. So what will be your approach towards the medicine? So the candidate just thinks for a minute, and then he tells, "Sir, I will tell myself I do not know." And examiner is very surprised. He says, "What do you mean? You are an MD person, now you will get a MD doctor. And if you are going to tell yourself you don't know, how you are going to face the patients?" Then he says, "No, sir, please don't mistake me. I have studied medicine for like the last 6, 7 years. You have taught us some textbooks, you have taught us some practicals, you have given us some anatomy practicals, how to dissect a human being. We know what are various parts of the human being. We know various diseases which you have taught us. We know the positive agents which typhoid is caused by salmonella, cholera is caused by Vibrio. This information I have got. I know that if the Vibrio patient comes to me, I have to give him electrolyte solutions to maintain his salt balance. If a typhoid patient comes to me, I have to give him some antibiotic. I know all this. But tomorrow, if typhoid comes in a symptoms which I do not know, typhoid presents in that manner, and if I don't tell myself I do not know, I am not looking for some new thing, then I will be stuck and I will never progress in my life. So I will have to keep on telling myself I do not know. And that is what, my dear students in microbiology, biotechnology, if you know, say that I do not know, there is always a chance you will learn something new. But if you say that I know everything, I have read few things and I am the master of the world, then probably that will be the end of your career. As far as the intelligence is concerned, you may progress as a human being, you may earn good money, but your brain will stop developing, and that is what I do not want. So the inspiration comes through famous scientists, the inspiration comes through books, inspiration comes, and then there has to be a motivation. The teachers are very good motivators. Unfortunately, many times I find that the teachers themselves are not really inspired or motivated. Maybe reasons I don't know. I don't want to list them. This is not a platform to go into the motivation of the teachers. But basically, I will like the teachers to, if they are listening to me, to be motivated. Finally, see, you are doing this. You are trying to polish a completely new generation, which is going to be tomorrow's citizen. And we all have to be very alert in telling what we are telling them and teaching what we are teaching them. So teachers, parents, and peer group. These are three motivating factors in a life of a student. The teachers, the parents, if they are listening to me, I have written one Hindi poem. Just listen to this poem. The name of the poem is Kayamat Se Kayamat Tak. जब पहुंचा हूं मैं उस मोड़ पे जिसे 12वीं कहते हैं, समझ न पाया था तब क्या क्या हो सकता है। पापा कहते थे बेटा इंजीनियर ही बनना है, अमेरिका जाकर घर ने वही बसाना है। खूब कमाओ बेटे, पैसे नहीं तो जिंदगी अधूरी है। क्या रखा है इस देश में, प्यारे सब घोटाला ही घोटाला है। मैं चुप था, क्या बोलूं मैं पापा के सामने भला, उम्मीद से है उन्होंने आज तक पढ़ाया लिखाया। मम्मी बोली, कुछ भी हो बेटे, लेकिन इंजीनियर मत बनना। अपने घर में कोई डॉक्टर नहीं है तो डॉक्टर ही बन। कितना अच्छा लगेगा बेटे तू डॉक्टर है बताने में, भाव बढ़ेगा कितना तेरा शादी के बाजार में। क्या मैं बोलूं मम्मी से, वो तो दंग उनके सपने में। मैं भी कुछ चाहता हूं बनने, कौन समझेगा मुझे। क्यों न पूछे कोई मुझसे, बेटा तेरा क्या इरादा है। लगता सबको अब भी ये दूध पीता बच्चा है। और सच बताओ, मैं नहीं जानता मुझे क्या बनना है। निर्णय लूं मैं कभी इतना सक्षम ही नहीं बना दिया। ये हमारी सिस्टम है। अनफॉर्चुनेटली मेनी टाइम्स द चिल्ड्रन आर नॉट रियली फोकस्ड, दे आर नॉट ओरिएंटेड। they come to they come to BSc because they have not got admission in engineering or in medicine. Nobody I have seen that has willingly come to, I mean, there may be few of them, but in general what I see is that they are forced to take the education. Now, forcing will never make a motivated student. And that is where, if that is where student will have to change. Suppose they don't have any option and they have to come for BSc and they are looking for microbiology. Now is the time. Forget about your frustration till they what you have experienced till today. Now is the time to begin a new life. You have got a very good subject, microbiology. You have got a very good subject, biotechnology. I mean, for me, the subject is not very important. That's what I told Valentina, that the physics, chemistry, biology, these are all the branches of sciences which we have made for our purpose. Basically, science is one single thing. Science is basic. If you look in your body, you have got physics, chemistry, biology, mathematics, engineering, how beautifully electrical engineering, electrical network is arranged in our work, in our body. Have you seen that? Have you seen the neural network in our body? If the neural network, it will beat any of the supercomputer systems in the world. So such a beautiful system consisting of all the science is there in our body. How we can say that we are going to restrict ourselves to a single subject? Well, the subject was there to begin with. It was a stepping stone in the field of science, and that field of science is so vast that then you can focus your approach. I mean, you can focus your approach, you can focus your attention and one particular subject. And once you achieve that, look for some other subject. This is what life is. That is what probably Newton meant, that when he says that a table is there, or he is looking at Hello.
Are you here, sir?
Hiya, na abhi? Can you hear me? Hello.
Yes, sir, you are audible.
Yeah. So I'll take up from where I left.
Yes, sir. Yes, sir.
Okay. So sorry for this break. What I was telling that we should we should understand what is our aim. What do you want in life? What liking you have? That is, they call it aptitude these days. And what is the scope? See, if you want to become a scientist, if you want to become a teacher, if you want to become an industrialist, if you want to become a farmer, you must have that aim very clearly defined in your mind. I remember in some of our school teachers were so inspiring. In seventh standard, Dr. Bhaba, when I was in seventh standard, Dr. Bhaba expired in an accident in Alps. His plane crashed, and that time our teacher had given us a beautiful lecture on who Dr. Bhaba was and what he has done for atomic energy. I don't know where he had all the information. He was an English teacher, but he gave us very good explanation on how Dr. Baba and how his team has helped India's nuclear program and how they have brought us at the level. That time Canada, the Cyrus reactor had just started in 1964, and Apsara also had gone. Apsara is a reactor. Apsara had gone critical, so we were quite getting advanced in that field. But of course, Pandit Nehru, our Prime Minister, earlier Prime Minister had supported this program, and through the vision of Dr. Bhaba, it was coming true. And suddenly he died. All this information I got when I was in seventh standard. There was no internet. There were not really very good books in our library, also the small town. But the teacher was a source of inspiration. So I decided that if I want to become somebody in my life, I will become a scientist. And well, I did become a scientist, and I never regret about that decision. Maybe if at that time I had got a job for in bank also, I would have reached some general manager stage of any bank. Probably that was in State Bank, and I would have earned far more money than what I have today. But I am very satisfied, and that satisfaction is what you want in life. So you have to decide. You have to decide what you want in life. Why you want to go abroad and you want to settle there, or you want to stay in your country and serve your country. Of course, by going out, you can still serve your country. I remember that shlok of Ramayana when Lakshman and Ram had won over Ravana. Lakshmana offered Ram that why not we stay in Sri Lanka, because it's such a golden city and such a beautiful city, and we have won it. So Sri Ram tells him, Api Swarnamai Lanka Name Lakshmana Rochate. Hey Lakshman, this golden Lanka is not attracting me at all. Janani Janma Bhumischa Swargadapi Gariyasi. My Janani, my Janmabhumi, Ayodhya, and my Janani, Kaushalya, Kaikeyi, they are all waiting for me. I will like to go there and I will like to serve them rather than become a samrat of this golden Sri Lanka. Now, the path is shown by Sri Ram. It's such an early stage in our philosophy. Indian philosophy is so beautiful, and Indian philosophy, see, need not be that every time you have to do sacrifice. Sometimes people say that by sacrificing you earn everything. No, you don't have to sacrifice, but you have to keep your thinking definitely open. That I will, because there may be a situation where actual sacrifice in what old terms, like say in our times, money was less. It was less with more than 85% of the people in general. So we had to live in something like poverty. We had to fight that unavailability of resources. Probably you people don't have to do all that because you have the resources available, you have internet available, you have your college book libraries available. But how many of you really read that college book libraries? I will like to ask a question to all of you who are listening to me that can you tell me in this lockdown how many new books you read? Probably the answer will be maybe few of you will answer yes sir, we read books, but generally the answer will be no. And that is what really bothers me, that is what is rather, and that's why we go in depression. If you take a book every week you will read one book. We had a nice school, and we had nice tradition in our school. Every classroom had 50 books other than textbooks in a small box like a harmonium box, and we were 50 students in each class, and our school used to be for 50 weeks. So every week every student has to read one book. That was the kind of unwritten rule. And by the time we left 11th standard, we must have read 350 books in our life, and probably that shaped our life. Sometimes people ask me, what is your inspiration? My inspiration were my parents. My inspiration was my school. My inspiration was my teachers. And inspiration was such a beautiful tradition of our Indian culture. All this is put together will make you inspired enough to be something in your life and try to achieve something, and make sure that whatever you do, you be a singer, you be a cricketer, you be a player. I mean, but you should try to excel, and that is what is will probably leave the footprints in your timeline. Otherwise, it will be like your Solomon Grundy, born on Monday, Christian on Tuesday, married on Wednesday, to kill on Thursday, died on Friday, buried on Saturday, and that is the story of Solomon Grundy. वो जन्म लिया, फिर खाना खाया, वो बड़ा हुआ, फिर फैमिली किया, बच्चे हुए, उसके बाद एक दिन शाम को हार्ट अटैक होके मर गया. ऐसा लाइफ नहीं चाहिए हमें. And to make this life better, your subjects like biotechnology or microbiology will make you see in your. I remember my mother, who was not really educated. She taught me difference between microbiology and nanotechnology. She had offered some kind of rava, shira, and laddu with the same amount of sugar, and then we found that the laddus which are having same amount of sugar as shira, that is halwa, what you call, was sweeter than the halwa. Laddus were sweeter than halwa. And at the same time, laddus can last long. Now, the reason for this is nanotechnology. When you make halwa, you put sugar in water, and this sugar gets dissolved completely. It goes in true solution. Now, when I ask students what is true solution, you must be able to tell what is a true solution. True solution is when the molecular size becomes smaller than that water molecule, then it becomes a true solution. It goes and occupies the intermediate space between two water molecules. The crystal structure breaks, and those sugar molecules occupy the intermolecular space in water, and that's why it becomes invisible. When that happens, the crystal structure is directly responsible for sweetness. So when it dissolves in water, the sweetness reduces, and that is where in case of laddu, you make syrup to make laddus, and in this syrup, you actually form a kind of nanocrystals. There is a recrystallization. When you make a very thick syrup, and then when the syrup is cooling down, there is a kind of fractional crystallization, and this crystallization will be in the form of nanocrystals, not in the form of original crystals. And nanocrystals, you know that they have got better qualities than the original crystals, so their sweetness will increase, and that's why laddus are sweeter. Now, when the microbes, now look for microbes. The microbes are not interested in sweetness. उनको नहीं कोई फर्क पड़ता ये लड्डू स्वीट है या नहीं है, उनको शक्कर से मतलब है. So to break that nanocrystal, they don't have that much of energy. So they never bother about breaking the laddus. So laddus preservation is far superior compared to halwa, which get destroyed second day, which gets rotten second day or third day, because bacteria can easily attack the dissolved sugar there, and they can take up that dissolved sugar faster because the molecular size is too small. They don't have to spend energy to break that, and that is what is called the science in kitchen, which was taught to me by my mother somewhere in 7th standard. So for me, if, this nanotechnology is never an very new science, because we have been living with this nanotechnology since the day we are born. You know, everything happens on in your body on nanoscale. You look at your the macromolecules, look at your cellular structure, look at your cell organelles. They are all working at nano range or micro range, and everything is happening. When a nano current, nano ampere current is then generated, it becomes microampere, and then you get the sensing in your brain that yes, something is happening, some electrical current is coming, and some thoughts are being brought to you through that current, and that's how the interpretation is happening. So this is what is finally you want in life. You want to make sure that you understand. See, like the happiness of understanding is very different. Once you understand a topic, you see the freshness and happiness on your face. If you don't understand the topic, you get strained, you get nervous, that how is that I cannot understand this, how is that I am not able to answer this. You all try to get bogged down, and then more nervousness comes. So it's a vicious circle, and that is where your experience, I mean, your aim of your learning should be that I want to understand the topic. Aim is not to earn degree. Well, degree is a side what you can byproduct. When there is a dairy, they say that milk is a byproduct, because in a cow, 20 years cow gives us gobar, and that is actually the product. Milk is only for 5 years, so milk is a byproduct. Similarly, you should look at the byproduct of your I mean, your gaining knowledge, your understanding knowledge, and that byproduct is degree. I'm not saying that it is not important. I will like every one of you to do PhD, every one of you to go abroad, see how beautifully science is being worshipped, how they are doing it. Now, take the case of today. I thought I will discuss with you some few aspects of viruses, because that's a very current topic. I'm going to discuss 6 different topics with you on 6 different days. Today's topic is the viruses, and because right now we are going through a very bad phase, the whole world is going through bad phase. My specific reference will be to the coronavirus. So when we are talking of the viruses, remember this Edward Jenner. He was that time smallpox used to be a killer disease, and the one smallpox epidemic comes, there will be at least a few hundred or few thousand people dead, and those remaining who have survived that smallpox attack will have either some kind of distorted face because of those pustules which have dried up on the face, or the eyes. These eyes used to get affected very badly. So the smallpox was one of the killer disease, and somehow the virus, bacteria, nothing was known at that time. This was, I'm talking of in 17th century. And Dr. Edward Jenner, he did first vaccine for smallpox, and to get that vaccine converted into the textbook, it took 100 years. But smallpox vaccine did come, and but before that, there was no word. The word virus came much later in Louis Pasteur. When he was talking about rabies, and he was trying to discover the cure for rabies. Made use of the word not virus, but he said there are some some organisms, some living beings which are smaller than bacteria, because Pasteur did know bacteria. At that time, bacteria were known. So he said there is something which is lesser, I mean, smaller than bacteria, which is responsible for causing rabies. And he did succeed in getting a rabies vaccine. But the word virus was used first by Martinus Beijering, or there was another Russian scientist, Dmitry Ivanovsky, in 1890s. Both these scientists, they found that the TMV, that is tobacco mosaic virus, was the responsible, and they showed that the cell-free filtrate, the filtrate was able to pass this bacteria, disease from one healthy plant to the other, and that's how the viruses were discovered. The Frederick Loeffler and Paul Frosch, they isolated virus of foot and mouth disease. So these initially these viruses were detected with plant and with animals, and the human being viruses came much later. But that's how the viral our understanding with virus, and then they started telling us that the virus is a link between living and non-living, and still we teach that yes, they are the link between living and non-living. So they why they are non-living? Because when they are outside the host body, they are called obligate parasite. Now, when a microbiologist is talking of the word obligate, obligate means absolutely necessary. So host is absolutely necessary for this virus to multiply. If outside the host cell, they will behave like a chemical crystal. So they can be crystallized. In fact, viruses can be crystallized. A Nobel Prize was given for that. This year's Nobel Prize also, if you remember, 2020 Nobel Prize is given for hepatitis C virus discovery. Dr. Altar, who was in Dr. Bloomberg's lab, and Dr. Bloomberg was another person who got the Nobel Prize for hepatitis B in 1976. That time in his lab, Dr. Altar was working, and he found that the whatever hepatitis B cases were not whatever cases were not attributable to hepatitis B, there could be a third different type of virus, and then he kept on discovering. So he and other two, then they decided the nature of the virus. They decided the Cox postulate. That's how those three people, Dr. Altar is one of the men of in that who got this Nobel Prize for this year. So viruses have always remained a center of attraction for all the medical fraternity, because these generally the concern is the virus means there will be some kind of disease, and it's not very wrong, because viruses are known to cause disease either in plants or in animals, including human being, or in bacteria. There are bacteriophages also, and these bacteriophages can be very dangerous, especially in the industrial scale. If any fermentation product is being made, if a virus attack comes, a good culture can get destroyed because of the viruses. So this is also these three types of viruses: the plant virus, the animal virus, and the uh, uh, bacterial viruses. They keep on cropping, and when you are talking of virus, when you are talking of bacteria, when you are talking of microbiology, you are talking of biotechnology, how your antenna should be. Your antenna should be very sensitive to the instrumentation which is with respect to these subjects. For example, microscope. Now, how many of the students here will be able to handle a good microscope on their own, or a good electron microscope have they seen? Now, this is it's not possible for everyone to see the electron microscope, especially at the small places. I am aware of that. But have you seen electron micrographs? Or have you seen what is the difference between electron microscope and light microscope? What kind of resolution we are talking? What kind of magnification we are talking? When a light microscope, what is the magnification of a light microscope which you use in your laboratory? Uh, can anybody tell? I mean, you don't have to really answer because we don't have a direct contact here, but just answer in your own mind that what is the magnification your light microscope has. If you are having a 10x or 15x as the eyepiece, and you have got a 40x as the objective, then 40 into 10 or 40 into 15, 600 times is your magnification. If you are using an oil immersion lens, the oil immersion lens has 100x, as the magnification, and the eyepiece is 10. So 10,000, I mean 100 into 10, that is 1,000 times the magnification will happen. So if your bacterial size is 1 micron, then it will become 1 into 1,000, that is 1 millimeter. And your eyes can really resolve, at least see 1 millimeter. Now, what is the resolution of your eye? Have you ever thought that question? What is the resolution of your eye? What is the minimum distance which your eyes can see or distinguish between the 2 points, the minimum distance? And many times students get bogged down by this question. They start looking blank. When it's a very simple thing in the third or fourth standard, at your student age, you start using scale. And I am sure right now you must be having scale on your table. Look at that scale. On that scale, you will find that there are 6-inch scale. If you have got, there will be inches on one side and centimeters on the other side. So look at the centimeter side. There is a centimeter, and each centimeter is divided into 10 parts. So each part is 1 millimeter. Now you can see between two lines. That means your eyes can resolve 1 millimeter. Now, if you put a exactly center spot between these two lines of 1 millimeter, you can still see the difference, and so your eyes can easily resolve a distance of 0.5 millimeter. But beyond that, probably it will depend on the eyesight, but maximum you can go up to 0.25 millimeter, but below that you can't. But your eyes can see smaller than that, because if you take a human hair, the diameter of a human hair is 0.1 micrometer. So you can see that 100 nanometers. So you can see that, but you cannot really distinguish between two sides of that. So resolution is not 0.1 millimeter, but definitely your resolution of your eye, you can say it is around 0.5 to 0.25 to 0.5 millimeter. Now, this is what you must be knowing when you are doing microbiology. This microscope, understanding that, why do you use a oil drop there? Now, for that oil drop, you must know some physics. You know what is the refractive index. The light rays are entering from bottom, from air to glass, and then there from glass to, again, if there is no oil there is a gap, there will be air, and from air to again glass. So that kind of movement, there is a lot of diffraction or refraction happening, and the light beam will not get concentrated. But if you have a oil there, oil has got the same refractive index, especially the cedarwood oil. These days they are using some synthetic oils also. This cedarwood oil has a refractive index of 1.5, which is same as glass. So that it becomes a continuous medium, and light coming from air, from bottom, then entering in glass, it will remain throughout in the glass, because then it will pass through the lenses. So there will be a concentrated beam coming inside, and that's why your resolution or your brightness will increase, and you can resolve it much better. And that is how the dark field illumination or the phase contrast microscopy will come in picture at later stage. But if you know this principle very well, I am sure you will be really inspired to use microscope, and you will never be afraid of microscope. Another problem, another thing which you should know about microscope is that the kind of fungal growth which happens on the lenses. Most of the these lenses, they have got a growth if you don't really maintain them properly. And that is where I find when I go for as an examiner for MSc these days, I don't go anywhere. But when I go there, I find that without putting any slide, you can see fungus in your lens. You must have also understood what I'm talking, because the lens will have a lot of growth on that, and there will be a fungus, and then to distinguish between what you are observing on slide on that lens, it becomes a problem. So maintaining that lens is also a part of microbiologist. It's also a part of your biotechnology, and you should not say that I don't know how to maintain a lens, because if you don't know how to maintain a lens, you are not a microbiologist, you are not a biotechnologist. Mind you, your instrument has to be extremely clean. How that genius of Antony von Leeuwenhoek, who is supposed to be father of microbiology, even at that in that 15th century or 16th century, he was able to develop the simple microscope, which was far superior to a compound microscope at that time. And who, none else than Robert Hooke had given that certificate that Leeuwenhoek's simple microscopes are better than what light microscopes, compound microscopes we are using. So that kind of lens, so that maintaining the lens, cutting of that lens, of course, we don't cut our lenses these days, but the maintaining the lenses is very important. And once you know that what is the resolution, what is magnification, and then you try to see your bacteria with this background knowledge, I think your eyes will be brighter when you observe the specimen. So coming back to these viruses, they are ultra microscopic. The sizes of these micro viruses can be in terms of nano. You know, there is a story of Pandora's box. There was a girl who was called Pandora, and one day when Farishta came and gave her some seal kiwi with ये देके गया with some kind of some packet which was completely sealed box, and this box was sealed, and he told he gave her a lot of gifts and told that don't open this box. And you know, if you are told that if you are told that you are you don't want to do something, you want to do it. If I tell you don't go in this room, your first thing will be that why sir is telling us not to go in that room. There is some secret. So you will, first opportunity, you will try to sneak in that. So Pandora also was like that, curious, and she opened that box, and the whole kind of negative germs and all disease-making bacteria and the protozoans, they came into the this world, and they started multiplying, and Pandora didn't know what to do, and that's how the diseases spread, and that's how the hell started appearing on the, of course this is a story. I have nothing to do with that story. So what I'm trying to tell you that all that understanding, just 1 minute. All that understanding will come once your concepts are clear. Okay, so the viruses are ultra microscopic. You need an electron microscope for that because they are in nano range. They are, see, average bacterial size is around 0.3, 0.4, 0.5 or 1 micrometer. If you look at E. coli, for example, it will be around 0.5 micrometer in diameter, whereas the virus will be 1,000 times smaller than that. So the smallest virus is around 20 nanometer, and the largest virus is in this world is around 450. Recently, somebody has reported a cylindrical virus which is more than 800 nanometer in length, and but it's so narrow, it is 15 nanometer in dia, but still the length is 800 nanometer. But generally, you don't find viruses in that range. The range of viruses is around 20 to 100 in most of the cases. Some of them are 200, 300, 50, but their number is less. These they don't live independent life. As I told, they are obligate. They will remain only in the body. They may contain either DNA or RNA. Generally, that if you look at the higher organisms, including bacteria, the DNA and RNA both will be present in a cell. But in case of virus, either of a DNA or RNA, the DNA can be a double-stranded or single-stranded. RNA can be a double-stranded or single-stranded. They are very host-specific attachments are there. You call it as uh, the entry points. Where it attaches, what kind of chemical composition is there, that is very important. And I think why I'm telling you why I began with virus is I expect a lot of things from you. I am going to give a lot of exercises, not in the form of writing to you, but I want you to educate, because you know that corona, I'm going to discuss that corona in detail in another few minutes. The corona is spreading like anything. Right now, it looks that there is a kind of dip in the growth. But if you look at America and Europe, the number is really increasing alarmingly, and World Health Organization has already told that the second wave could be very dangerous. It could be more killer than the first phase. So all of us have to be alert, and all of us who are microbiologists will have to contribute to the society by spreading the correct awareness. Right now, I still find people who are moving around without masks, without, they are not even bothered about the severity of the disease. They are not probably aware. Most of them are not aware, even despite of so much of talk on TVs and various channels. People are still not aware. If you look at the now Diwali crowd will start, and the type of people, I mean, the way people are moving in various cities and not maintaining any social distance is going to be very, very tough if the second wave comes, because medically we have seen how bad we are, especially the public health facilities which are there in our country. And so it becomes all the more responsibility of the microbiology and biotechnology students. And that's why I chose today Corona as my first topic, so that in next 6 days, I will get the feedback from you, what kind of community awareness, what kind of measures you are taking to stop the second wave or to stop the coronavirus from your locality. And just imagine, just remember one slogan, that if you keep your family free of corona, India will be free of corona. Because each family decides that my family will be free of corona, the corona will not spread. And once the corona chain is broken, it will not be able to spread it very fast as it has done, because of the chain is not broken, chain is continuing. And I'm going to discuss all those scientific aspects in a very short time. So please remember, that's why I chose this topic today to talk to you as corona, and there is a lot of scope for R&D also. I am going to talk about that also. You must keep on reading some of these papers which are coming, the bradykinin storm, which is happening because of the coronavirus in your body. Various bradykinins are getting secreted in very larger quantities than normal, and what kind of havoc they are causing, because then they will actually divert the all cell attention, and the senses will get lost. And that's how people are losing senses, that even the oxygen, if it is less in their body, they are not able to, that is called happy oxic state. uh, they are deoxic, the oxygen levels are definitely less, but it is not reflected on their face because the sense is lost. And that's a very dangerous thing, because suddenly a person who is moving around and has a 80% of CO oxygen or 70% of oxygen in his body than we expected of 95 or 96, he may suddenly collapse and he may die also. So all this can happen, and that's why awareness of corona and spreading of it and telling them properly about getting excessive fear is not good, but at the same time, you cannot take corona lightly. Although it's a small virus, it has really brought the entire world at halt. The whole world has completely stopped functioning. You have seen, we have seen that from March to July, what happens in our country, and it's such a bad situation. So let us first understand the what are our uh, uh, what is in our scope, what we can do in our college as research. We can take up some small projects of research, simply that RT-PCR and those machines are available to all of you. So if you are going in your city, say Rajkot, Valentina is in Rajkot, or people are listening to me, say from Mumbai or from Pune, all of these cities, they have got good corona spread in their cities. I don't know about Rajkot, but Pune and Mumbai has plenty of that. In a given criteria, in a locality, if we take sewage sample and check for RT-PCR and see the number of viruses are, the corona is positive there, then possibility is that the incoming 10 days or 15 days, there will be an outbreak of a small epidemic in that area. that's an indication. So all such small projects you can take up, and RT-PCR is, I think most of these colleges also are having RT-PCR, or universities definitely have RT-PCR. You can take these projects, small projects, and be helpful to your society. The virus particles, about the size, again, if you want to compare, in one single bacterial cell, you can have as many as 2,000 viruses. 2,000. And if you look at a human cell, average human cell, like say red blood cell, or you take any of these squamous epithelium, you take any of these muscle cell, you will find that as many as 50 million viral cells can multiply in that. Once the particle enters and takes over the cell machinery, the number can go up to 50 millions. And when this number becomes, this is called as the outburst will happen, and these 50 million particles, if they are getting released into the body, I think God only can save you. So that's basically the difficulty in this, that you have to restrict these bacteria, I mean viruses, from entering in your mainstream and then getting established there. The coming back to the structure of the virus, there are such a simple organism. I don't know how God has made it. One research topic I want to give you is how can you really predict how these viruses have come onto the earth? They say that the beginning of the earth, or at the time of when Big Bang, there was no life, but the first life came into existence. Was it virus or what is the bacteria? That's the question I want to throw in front of you, and try to get an answer to this question, that and try to give a good legitimate essay on that. If you can share this essay with Valentina or with me, we will give our email address at the end, and I'm sure you must be having our email addresses. We can even think of giving a prize if there is a very good essay on this, that what is the origin of viruses really. And this is a very simple structure, how life can be, simple, how life can be complicated. There is a king who has got a palace of more than say 100 or 200 rooms, and to maintain that palace, then he needs a very big machinery. Whereas there is a small in Jhopda and hut, and in this hut there is a person who is very satisfied, who is staying there. So both are possible. So similarly, this virus is which has got only two things. It has got a protein coat and it has got a nuclear core. This nucleus has, nuclear core, as I told you, it can be a single-stranded DNA, or it can be a double-stranded DNA, or it can be single-stranded RNA or double-stranded RNA. Viral genome is very small. HIV virus has only 9 genes. HIV virus has only 9 genes, whereas the largest virus, which is called the Pandora virus, it has got 2,500 genes. Now, if you want to compare with E. coli has 4,000 genes, and the average human cell has about 21,000 genes. So compare virus with human cell. 21,000 genes are there in your cell, and there are 9 genes in HIV. In coronavirus, I really don't know how many, but they will be in terms of in 2-digit or maximum 3-digit. I don't know even 3-digit, but they will be in 2-digit. But see the dominance. The dominance of coronavirus over human cell, which has got 21,000 genes, and that is something which we must understand in this world. You can really correlate with so many things. You need not have all the time the large as the unit for better. Large need not necessarily large is better. The small can also be better. The God of Small Things. There is a nice book by Arundhati Roy, or the God of Small Things. So similarly, this virus is I can call it as the master of small genes. It's a very small number of genes. It can actually dominate huge organisms like human being or This coronavirus has affected certain lions also in some of the zoos. That is the report now coming, that many animals are getting affected by the coronavirus, which is right now present. And this coronavirus is this is novel coronavirus, COVID. So the viruses actually we are always looking at the destructive. Some of them are really good. The beautiful tulip colors. The Netherlands is the country which is very famous for beautiful tulips. And in Netherlands, they found that in somewhere about 100 years ago, this color of tulip is not actually the natural colors. These are due to the infection of viruses. So these viruses infect the tulips and they alter the plant metabolism in such a manner that the petals develop beautiful colors. So the viruses at least in tulips are not really destructive, but they have offered us the very nice colors tulips. The extreme habitat in Antarctica, you will find that these viruses are present in a very small sample there. They found 10,000 different unique viruses. So there are unknown places, and that is where I don't know where I read. I can't give you a reference, but when I was young and I was studying MSc, we had a teacher called Dr. Y.S. Kulkarni. He was a very nice person as a soil microbiologist. He had mentioned sorry, there was a break again. Can you all hear me? Can you all hear me?
Yes, sir. Yes, sir.
Yeah. So there was a break. Sorry for the break, and there was no advertisement. So you can consider that I was advertising for Corona mask. You should use mask. Okay. So that was my advertisement for this break. Coming back to what I was saying, that in Antarctic, they have found 10,000 new unique viruses. Ocean biodiversity, you know what is the ocean biodiversity as far as virus is concerned? If you are looking for ocean water at certain places, you will find 1 million bacteria per milliliter. But in the same place, you will find bacteria, I mean viruses, 10 million viruses per ml. That is the kind of biodiversity of viruses. And they are only, we know the 20 families of human viruses and probably some few viruses, a few families of bacterial viruses and few families of plant viruses. So we know very little of viral world. We don't know how these viruses are going to, and that's why these researchers come with papers that be aware there is going to be a new virus, there is going to be a new virus all time. You will, and that's where you should keep a tab with the advances in microbiology through the journal papers. These huge number of viruses are responsible for controlling population of eukaryotes and prokaryotic cell, and they are also influencing the biological activities like photosynthesis and the various elemental cycles. The viruses are now useful. I mean, human being is very good in making use of even destructive policies. And this virus are less harmful viruses like Vaccinia or adenovirus. They put a genetic material from pathogen like HIV and they use it as a vaccine. So these are called as the very Vaccinia vaccines. These are where actually you are putting it offers you beautiful immunity without actually causing the disease. So this is a new kind of vaccine which is coming up. This is called Vaccinia vaccine or adenovirus vaccine. These genetic diseases are also like cyclic fibrosis or the sickle cell anemia. There you put a normal gene from a normal individual and put it in adenovirus, and this vehicle you put it in a person who is having this genetic disease. Probably that genes will get incorporated into his body and he may be cured of these genetic diseases. So that kind of approach is also being tried. Phage therapy is being tried to control the bacterial infections, especially in food. The two dangerous bacteria in food are E. coli and Listeria monocytogenesis. These are very common in meat and poultry. So the USDA, United States Department of Agriculture, has given clearance to the use of such viruses. Now look at with this background, look at coronavirus. The pandemic was declared in 11th of March, and today we have got more than 4 crore 67 lakhs of people infected with this coronavirus. Out of that, 3%, that is 12 lakh people have died. In India, we have 82 lakhs, more than 82 lakh people who are affected, and 1,22,642 people, which is around 1.5%. So this is definitely a low, they call it as the low fatality rate. 3% or 1.5% is low fatality, but mind you, that should not be make us lethargic, because those who suffer, they suffer really very badly. And we should not be in that rank who our immune systems, everybody's immune system is not necessarily good. So we will have to check that we keep this infection away. Now, what we know of coronavirus is a very large family of viruses. And these earlier two viruses, MERS, that is Middle East Respiratory Syndrome and Severe Acute Respiratory Syndrome, that is MERS and SARS. These were the two viruses which were very fatal in earlier days. Now this SARS
Hello, everybody. It was really very interesting lecture, and we were really absorbed in this talk completely. We have seen so many different aspects of microbiology and biotechnology, also nanotechnology from Sir, and he knows too much about microbiology, and we hope that every day we are going to get more and more from him and from his experience. So we are waiting for Sir to be connected. So let us see. I heartily welcome our trustee, Veena Madam, Veena Bin Pandi, and Dr. K.K. Khakkar Sir. They both are with us. So it is our privilege to have them with us. And we are very much thankful to you that you are being with us. Thank you so much, ma'am and sir. We hope that you like this lecture series and you will come to know very new things every day. When we read the books, it will be not so much interesting and not so much revealing the truths. But when we listen it from somebody who is experienced and who has already digested everything, so it will be really very interesting and beautiful to listen to them and to understand the things properly. So each and every concept you try to learn from Kale sir.
Yeah, madam, I will. I joined again.
Yes,
I
sir.
will just
Welcome,
finish
sir.
it, my last line, I will finish it so that you can take over again. Okay. I'm sorry again for this break. I don't know why. I have never experienced this break in last 6 months. I am doing a lot of meetings. Today there is some problem with our internet provider, and this is certainly not from organizer side. It is from my side which is happening. So I regret this problem, and I sincerely uh, accept to. I mean, take this job with my internet service provider. Tomorrow I will make sure that such kind of break is not there. So what I wanted to tell you that the COVID peak is not over. COVID is going to be there for some more months or probably a year. Vaccine is going to be very difficult because this vaccine is not of you must be seeing so many good news, bad news, good news, bad news about vaccine. ये fail हुआ, ये fail हुआ. There are 10 different vaccines which are being tried in various parts of the world, but there was a lot of talk about Russian vaccine. Now nobody talks about it. So you know that vaccine is not going to be there. So basically, you will have to maintain move with mask. Never forget to go out with mask and make sure that 500 people you tell every day, and that is how the message has to spread. How you make that 500, it is your job. You make it 5,000, I'll be happier. If you make 5 lakhs, it will be still better for the country. But make sure people move with mask, and after coming home, take steam, and whatever virus gets lodged in your throat, you should be able to remove that. Tomorrow I will talk about it in little detail in first 10-15 minutes and then take up the new topic. So thank you very much because time is getting over. Thank you very much, organizers, and tomorrow let us hope that it will be an undisturbed lecture.
Thank you very much, sir. So now, all of us, we are really very much enlightened by sir talk given by Kale sir. Now we are going to meet tomorrow sharp at 2:30. So please wait for him to come and connect in the time. So we are going to learn something very beautiful things tomorrow again. See you and take care of yourself. And Jai Jalaram and bye to all. Thank you so much.
Thank you.
Okay, thank you very much.
Thank you, sir.
Thank you, sir.
Thank you, sir. Thank you very much. Namaste.